Okay. All right. So now this is the endospore stain. Now with the endospore stain, endospores are very heat resistant, dryness resistant. They are resting, kind of called resting cells, but they're not technically cells. Basically, bacteria that produce endospores, if they're in an environment where the cell itself starts to die, so they run out of water, they're starting to get old, basically they produce an endospore, which is basically just a wall around DNA. And that endospore can sit for like up to 30 years in soil or something like that. So some bacteria that produce endospores are bacteria like that causes Clostridium. Okay. Now, with the endospore stain, in order to get it to penetrate the stain, we have to use heat. That's why I have the boiling water. And it's not going to boil for very long because it's unplugged, but I have it boiling just to demonstrate. So now we're on page 49, and then if you look on page 50 is the procedure. And now with this procedure, it says to draw 3 circles and heat fix your bacillus first. So we've made our smears, we've heat fixed them. The other procedures we were doing today, we didn't heat fix. Okay, so we have our cultures fixed on here. And we're going to have 3 different kinds of bacillus. One that's normal, one at 24 hours, and one at 72 hours. And we're going to see if we see a difference. Now for this particular procedure, we're going to put it on this boiling water, and you can maybe still see the steam, but imagine it's continuing to boil, and we're literally going to steam it, okay? This is so that we have the heat so that the stain can penetrate, okay? I'm going to take my goggles off. I'm getting overheated here. All right, so now we have the boiling water. We have the slide on it with our fixed stains, our fixed smears, not our stains, our fixed smears. So the primary stain is malachite green. If you take a look on page 50, you'll see that the first procedure says to put a piece of paper towel on it. I don't want you to do that. I don't think it's necessary. Basically, that's just to keep it from evaporating because, of course, when you heat liquids, they start to evaporate. What I prefer is just you keep an eye on it as it's steaming, and you just keep adding the malachite green if it looks like it's starting to dry out. Okay? So skip the paper towel. So now it says to cover with malachite green. Okay, so I want you to add the stain after you have it on the beaker of water that's boiling. Okay, don't put the stain on first and then try to move it to the beaker of water. Okay, that's just messy. Now it's going to be steaming for 5 minutes. Of course, I'm not going to wait that long during my video, but as it steams, you take note. You can see maybe it starts to dry out. You'll add some more malachite green. You don't want it running over the sides into the water, but just keep adding it just to keep it moist. If you don't keep it moist and it dries out, it's not going to stain properly. So we're heating it so that the stain will penetrate the spores. Okay. Now this is going to be kind of hot, so I may want to be careful. So I'm going to go ahead and just use a clothespin to grab onto it, or maybe not. Okay, and so I'm going to take it over here, and I'm going to rinse it. All right. Now for this one, the counter stain is saffron in, just like it was in the gram stain. So we're going to add our counter stain for our 5 minutes, or not 5 minutes, for 1 minute. Okay, and then we're going to rinse that off. Okay, and then we would blot that dry like we've been doing with all the other ones. So that was the endospore stain. Now one thing I want to just point out is when we're doing all these stains at one time, you're going to have to be very organized, and at any given time you may have 3, 4, 5, 6 different slides that you're working with. So just make sure you keep yourself well organized as you're doing all these procedures.